Now let's talk about vesicular transport. Vesicular transport is where you move substances across the cell membrane, and you sort of use the cell membrane to package things and swallow things for you. The cell can swallow stuff like endocytosis, and it makes a little indentation in the cell membrane and then makes a little vesicle. This is endocytosis. Exocytosis is where you have a vesicle over here surrounded by a phospholipid bilayer, and these vesicles would fuse with the membrane and release their contents to the outside. So here you're using the cell membrane itself like a packaging system over here to absorb or to release certain substances. So now if we look at the this one and the two divisions, endocytosis, you are swallowing stuff into the cell or you are drinking stuff into the cell. So under endocytosis we have some subdivisions. Let's just get rid of the exocytosis part right now. So you are internalizing substances via the cell membrane itself, using the cell membrane to encapsulate the substances you want to swallow and take it in. So there is something called phagocytosis and pinocytosis. Phagocytosis is what they call cell eating. Okay, cell eating. And pinocytosis is cell drinking. It's based on the size of the molecules that they swallow. Cell drinking is more for water and some small molecules with that. Small molecules or ions. Pinocytosis is like a small version of phagocytosis over here, okay? And so it just absorbs stuff. And an example of this is how fluid is absorbed by the endothelial lining of the capillaries. So these are your capillaries, they have endothelial cells, and a good portion of water is absorbed through pinocytosis. It absorbs water into the cells through pinocytosis over there, okay? So that's one of the mechanisms over there, through the endothelium, as a good example for pinocytosis. Phagocytosis is cell eating. Cell eating. So what happens is you see a big chunk of molecule, maybe a piece of bacteria or an antigen that is coated by antibodies that gets swallowed by the white cell, usually, as an example. So this is your phagocytosis. I'll just talk to you a little bit more about phagocytosis. So if we look at phagocytosis, like I said, the best example is a neutrophil or a macrophage. Neutrophil or macrophage, WBC cell, that is going to engulf a sort of microbe like a bacteria, a certain antigen. This bacteria could be covered in certain antigens all over it. These are molecules that elicit an immune response. That's what an antigen is. Okay, and your immune system eventually learns how to fight these different antigens, and they make certain substances. One of them is an antibody. So this is an antibody. It's a Y-shaped little molecule that I'm drawing here. You get different types, IgG and IgM. These are the main varieties that help in combating most of these invading pathogens in your body, these microbes. And first you make the IgMs, and then after a week the IgGs come into play. That's how it is. When you get an infection, the first antibody that comes around 5 days after the infection is IgM, and then after some more time, like after a couple of weeks of the infection, eventually the class of antibodies that are created are IgG. So this is called class switching. Anyway, so the antibodies will come under the heading of acquired immunity, and here under acquired immunity you see these antibodies being produced, and they will coat these white cells over here. You could see They are coating the white cells over here because they are sticking to these antigens. Now, what do these antibodies do? There's also a complement that gets involved in this, but let's just focus on the antibodies. So these antibodies, they bind to the antigen and they sort of act like garnish or spice when you put some garnish or nice mozzarella cheese on your pizza. It makes the bread look more delicious. You want to eat pizza instead of just plain bread. So that's what your antibodies do. They coat these microbes, and this process is called opsonization. The opsonization will coat your antibody, use the antibodies to coat the microbe over here. Now what happens is the white cell is very much attracted to this portion of the antibody. You see this antibody over here, this part is called the Fc portion, also known as fragment crystallizable. Right? And we'll talk about the structure of the antibody and why it's called that. Look, this is the antigen binding site. Here and here. And this is the Fc portion over here. This part is the Fc portion over here. So what happens is you have your antibodies that bind over here, and the Fc portion is present over here outside. And this is very, very attractive to your white cells. Your white cells have receptors located on their membrane, what they call like coated pits. There are these pits over there which are full of these receptors that are very sensitive to Fc portion of the antibodies. So these are your coated pits over here. Coated pits. Okay, and what happens is these receptors bind to the Fc portion, and they realize that there is something very delicious to eat over here. It's sort of like the antibodies bind to the antigen, and they also say, hey, look, I got something here. So these white cells are sort of short-sighted. They don't see so far. But once they get hold of an antibody, they realize something delicious is here, and they start to grow and swallow up this entire thing. You can see it's like a big monster here, just like Pac-Man trying to eat everything over here. So this is how your white cells swallow stuff. We'll just talk a little bit more in detail about how this works. So if we look at the antibodies, we have that coat the antigen or any substance that has to be phagocytosed. We have a cell, and we have receptors over here. These receptors are located on what we call coated pits. Okay, there are some pits, some little sort of depressions here, little slight depression in the cell membrane with the receptors located over here. Now these receptors can bind to Fc portion of a certain protein or anything for that matter. Now I want to draw this neatly, so I just want to make this extra neat. Okay, we could do better. Yeah, this is better. Okay, I need a little bigger one. Okay, so now at the end of the receptors, we'll just label this as the receptor. The receptors are here. And what happens is at the bottom of these receptors, there's an important molecule attached over here. We call this adaptin. And what happens is when the receptor binds to a certain antigen, when you bind to a certain antigen, let's just draw these little triangles, little nacho chips looking antigens, because nachos are quite delicious over here. So they bind to these little receptors. This activates the receptor, and adaptin is bound to these receptors. Now what happens is the adaptin is then going to allow another important molecule to stick to it, and this is called clathrin. Now look at the structure of clathrin. It's got 3 legs. They're called triskeletons. Alright, so that's the structure. Well, this clathrin organizes itself and attaches to these adaptin molecules, and it causes the cell membrane to slowly get bent inwards. It pulls it in. So what was straight like this eventually bends inwards because the clathrin is sort of pulling it inwards over there. Okay, the clathrin plays an important role in this. So the clathrin coated pit is formed with the help of the adaptin that's linking and bringing everybody together over there, and this clathrin is going to pull this membrane inwards and inwards like a sort of a bag that's being with a portion of it just going inwards, sinking into itself. And eventually you get this sort of an organization where the receptor will look something, the membrane starts to look like this, with those receptors bound to those antigens over here. And of course we have the adaptin over here, and the clathrins doing all the muscle work over there, where you have your 3 legs over here, 1, 2, 3, stuck around the core molecule. So think of a pelvis and 3 legs coming out of it. And eventually what's going to happen is the neck of this little depression, it's going to come really close. The 2 layers of the bilipid layer are going to come very close, and then a special molecule comes over here, and this molecule, which is going to just come over here and nick everything and make a little bag, we call that dynamin. Dynamin. This is a guanosine triphosphatase type of an enzyme. It's a molecule with GTPase activity, so it uses GTP for its energy, and it breaks down the GTP molecule. So it's got GTPase activity over there, and it's going to just chop everything and make a nice little vesicle over here. So from this we get this, and finally the dynamin comes and it separates the little vesicle. It just nips it like that, like when you're making mozzarella balls or certain types of other dishes, where you take a little bit of cheese in your hands and you just plop it off like that. If you're making a certain shape, or even certain doughs or something with the bakery, you could just associate with that. So now we have the receptors that have been internalized over here. Right, these are the receptors which we've internalized over here, and the clathrin, eventually it says, okay, my job's done, I'm just going to dissociate from this place. So it will just break off and it will recycle itself again and bind to the next bunch of receptors over there. So what was a coated vesicle eventually becomes uncoated. So there was a coated vesicle coated by the clathrin, and now it is uncoated because the clathrin leaves. Now it is uncoated. So this is how your endocytosis happens, and eventually after the endocytosis happens, like it's absorbed, that microbe or that antigen or anything, even something that has to be digested for that matter, what's going to happen is eventually this organics molecule has to be absorbed and destroyed and digested. So lysosomes are supposed to come over here and fuse with the vesicle that you absorbed, that you just got into the cell. So the vesicle you just got in, we call it a phagosome. And when the lysosome comes with all these enzymes, it will fuse with the phagosome, and all these enzymes are going to start chopping every big molecule, organic molecule that it could find. So it makes something called the phagolysosome. So this is how phagocytosis happens. And when it comes to pinocytosis, a similar thing happens, but it's on a much smaller scale. So that's how your vesicle is going to work over here. This is your endocytosis in a nutshell. Right? You must keep an eye on the interplay of different molecules over there. It's not the simple amoeba that's engulfing something. You can't talk about that here. You are supposed to focus on how the neutrophil or the macrophage would engulf different antigens over here and swallow everything up. Using the help of certain molecules like adaptin, clathrin, dynamin with its GTPase activity that plays an important role over here. And the next thing is exocytosis. Now exocytosis is the opposite. Here you have a cell, and the cell has something coated in a vesicle. The vesicle looks like the cell's bilipid layer, the cell membrane's bilipid layer, and it eventually goes to the cell and it fuses and it removes its contents out into the extracellular fluid. So this is how exocytosis is supposed to work. Now we'll go into more details over here. Where do we need this? There are for example, neurons that release neurotransmitters when an action potential goes to the end of the neuron. So there's the neuron, there's the axon, and there is the terminal boutons, the ends of the axon. So we get these branches, up to 1,000 branches sometimes from one of these neurons. That's the cell body. And at the end of the cell body is this branching. We call this terminal arborization. Terminal arborization. And this terminal arborization, there are branches up to 1,000 in number sometimes, and at the end of this it kind of looks like a button that's tied to a little string over there, that's stitched into your shirt or your coat. So this is where the synapse happens, and in these little synaptic knobs or synaptic boutons, I shouldn't say knobs, synaptic boutons, it's French for button. What happens is it can bind to the dendrites. Dendrites are, projections of cell bodies over there. Sometimes directly to a cell body of another neuron, for that matter. So this could be the next neuron over here. So this is your rudimentary diagram. So here, release of neurotransmitter happens through exocytosis. Other examples are certain cells that secrete hormones. Your endocrine cells. Endocrine cells. So we have neurons that do this. And you have endocrine cells that secrete hormones throughout different parts of the day, sometimes throughout the entire day, like thyroid. However, these hormones are released under certain stimulation. So that again requires exocytosis. Then you have something called your fibroblasts, for example. Fibroblasts. Fibroblasts release collagen. This makes the ground substance for your tissues and lends structure to a lot of tissues in the body. And that's very important again, not just for the maintenance of tissue integrity. So collagen plays an important role. They're present in the bone and the cartilage and the basement membranes of different cells. So now how exocytosis happens, I'll just give you an example. In the synapse, for example, an action potential comes down along the axon, and when we reach the terminal bouton, what happens is there are special channels for an ion called calcium. So calcium, voltage gated calcium channels. Voltage gated calcium channels exist, and calcium enters into the cell. Now remember, most of the cells over here, whenever they have to release something like a neurotransmitter or a hormone, calcium has to enter into that cell. Then these vesicles present in the cell containing the hormone or containing the neurotransmitter will be motivated to fuse with the cell and undergo exocytosis. But that happens when calcium enters into the cell. There are only two exceptions that I could think of where calcium not entering causes secretion, but everywhere else calcium entry causes exocytosis in the body. Exceptions are parathyroid hormone gland, PTH, the chief cells in the parathyroid gland, because these cells secrete calcium. These chief cells secrete parathyroid hormone when the calcium becomes low. That's how they're trained, because parathyroid is supposed to increase calcium levels when it goes low. Okay, and another thing is the cells that secrete renin for the renin, angiotensin, aldosterone system. This comes in blood pressure regulation. I'll have a video of all of these out soon enough. So calcium entry causes exocytosis, and these vesicles, they fuse not just blindly with the membrane like so. What happens is, if this is the membrane, there are certain molecules here on the membrane, and they're called T-snare proteins, target snare proteins. And the vesicles, they have some receptors on them called V-snare. I should write that in capital. V, vesicular, S-N-A-R-E. And you have these acetylcholine molecules bound over here inside them. Now the V-snare and the T-snare, they are going to fuse and fit like a lock and key. And that is going to cause the exocytosis. Okay? Can exocytosis in the neuron happen without V-snare, T-snare interaction? Yeah, those are called kiss and run discharges. Kiss and run. Yeah, it's funny. Physiologists have a good sense of humor. So sometimes they do happen, but they're not that effective in creating much of a physiological action in the body. They are picked up by certain electromyographic recordings, but not commonly recorded so easily over there. Okay, so you have the snare proteins that are important in this. Well, exocytosis happens because even in hormones or fibroblasts. And how does this happen now? So you should understand that like certain hormones are polypeptides, and like insulin or glucagon or growth hormone. And if you know proteins from cell biology, they are made from, if this is a cell, there's a nucleus, and the nucleus has DNA. DNA has the blueprint for all proteins necessary for life. So the DNA gives rise to mRNA. This is called transcription, and the mRNA has the blueprint which the ribosomes and the different amino acids come together and make into proteins. I'm just giving you a simple example over here. Now these proteins, where are they? They are made, and then they are going to go to the endoplasmic reticulum of the cell. ER, endoplasmic reticulum. Right, they're like little stacks over here, and after the ER there is something called, let's draw in a different color, make this a little bigger, this cell. Yeah, that's better. So we have ER, and now I'm going to draw Golgi apparatus over here. So we have something called the Golgi apparatus. This is all important in the packaging of substances over here. So this is Golgi. So the proteins are made, they go into the ER. This is the protein I'm drawing in red. They enter into the ER, they get modified, and then from there they go into the Golgi apparatus through what we call the cis part of the Golgi. They get modified, packaged, and then they come out, they bud off, they move through the cis, and then they reach what we call the trans part of the Golgi, and they bud off from the Golgi over there. So we get these little buddings of this, like little vesicles budding out, containing that substance which has to be secreted. Not just endocrine stuff, any thing that has to be secreted. So now they are existing in these vesicles over here that you could see. These vesicles I'm drawing over here. These have the thing that has to be secreted. Now, if calcium is going to enter the cell under the influence of whatever is regulating it, or calcium can also be released into the cytoplasm from the endoplasmic reticulum itself, smooth endoplasmic reticulum especially. Smooth ER releases calcium because it stores calcium. So again, calcium levels rise, and this increase in calcium is going to cause these vesicles to fuse with the membrane and release substances out. This is how these things work over here. It will release the substances out over there. And so the point I want you to say is to understand this calcium comes in and then stuff goes out. Exception is parathyroid hormone and the renin-angiotensin-aldosterone system. So the exocytosis happens through two styles of pathways. One is your constitutive pathway, and the other is the non-constitutive or falsely named regulated pathway. What happens is both the pathways are regulated. Now I'll just talk to you about what is constitutive versus non-constitutive. What I just explained to you, like a hormone is produced from the DNA blueprints, the mRNA, the protein, it goes to the ER and then gets packaged into vesicles, as an example. Now what happens is some stimulation comes from a higher place, like the hypothalamus or pituitary, and that gives this cell the signal to release the hormone. So only when it is told to release, it releases. This is called non-constitutive. You understand? It only secretes when it is supposed to secrete. While constitutive is in the case of production of certain antibodies or collagen from fibroblasts, these cells just keep secreting them, keeping a certain level in the body. So this is always being secreted. It doesn't need a special signal every time, but in non-constitutive there is a special signal that comes and causes the secretion to take place. Okay, so this is a very, very simple rudimentary understanding of this. There's much more you can read about, but it's not what we'll be going into so much detail right now at this point of time.